Hello, everyone, and welcome to today's webinar, One Health Genomic Surveillance Simplified, Illumina Microbial Amplicon Prep. I'm your host, Christy, and this LabRoots educational web seminar is brought to you by Illumina. Before we start, let's get you acquainted with the webinar interface. Resources. Here, you can access additional information, including an application note, a link to the Dragon targeted microbial app for data analysis, and a brochure showcasing Illumina's infectious disease and microbiology solutions. Q&A. Have a question? Type it in the Q&A box and click submit. We'll answer as many as possible live, and any remaining questions will be addressed in a follow-up resource. Survey. Please take a moment to complete the brief survey. Your feedback is vital in helping us provide you with the most relevant technical resources following the webinar. Now let's get started. I'm pleased to introduce your moderator for today, Darcy Whitlock, Associate Director of Global Product Management for Infectious Disease and Microbiology at Illumina. Welcome, Darcy.
Great, thank you. We saw during the COVID-19 pandemic the importance of genomic surveillance of pathogens. And public health labs worldwide have continued to expand this type of surveillance to other targets. Today, we'll hear from scientists at two pioneering public health laboratories, the National Institute of Virology in India and Fiocruz in Brazil. They've applied their experience from the pandemic and their expertise in genomic surveillance to address a broad range of public health challenges within the One Health framework. I'm excited for them to share their experiences with you today. Pathogen surveillance and pathogen genomics is being increasingly integrated with routine surveillance for communicable disease within human populations. Genomic surveillance improves the understanding of disease transmission, especially from non-human populations like zoonoses. It can help identify mutations and new variants, and surveillance can also help monitor the impact of new variants on vaccines and treatment efficacy. And we found that targeted surveillance of single pathogens really simplifies and speeds surveillance compared to using large panels. And this all helps improve public health response. Again, during the COVID-19 pandemic, that really highlighted the importance of targeted genomic surveillance. We saw heightened public health responses when new variants came out, like Delta and Omicron. We've seen continual vaccine updates in response to new mutations and new variants. And it really brought a big spotlight to the value of wastewater surveillance for monitoring communities without regard to people's access to healthcare. Genomic surveillance has continued to impact public health responses in 2022 and 2024 during the Mpox global health emergencies. It also played a large role in monitoring the highly pathogenic avian influenza global outbreak, the H5N1 strain. And it's very helpful in arbovirus differentiation, looking at things like dengue and chikungunya. And with those single target, single pathogens, Amplicon next generation sequencing is ideally suited to that. A lot of labs are already familiar with Amplicon-based sequencing from the pandemic using COVID-seq. And the Illumina Microbial Amplicon Prep that we'll be talking about today is the same chemistry as COVID-seq, but really expands its usage to any other virus. It allows for custom primer designs. And if you're looking for primers for a specific virus, Primal Scheme is an ideal source. It's a website you can go and design your own primers for your virus of interest, or you can check out what's already been published on locations like protocols.io. The IMAP kit works with DNA, RNA, or total nucleic acid. It's really variable for genome sizes or the number of amplicons you're using, and very, very flexible for sample types. We've seen everything from nasal swabs to wastewater perform very well. It also has very simple bioinformatics. You don't have to be an expert to use these products. Basebase.com allows analysis for both predefined references, and also you can very easily upload your own custom primers for analysis. And just a point, there's a lot of literature out there talking about COVID-seq and modifying that for use with other viruses. Any papers on COVID-seq also apply to the IMAP kit. Our first speakers are Dr. Varsha Potdar and Dr. Pragya Yadav from the ICMR National Institute of Virology in India. Dr. Varsha heads the influenza group at ICMR-NIV and plays a key role in shaping India's public health strategies for respiratory disease. Dr. Pragya is a senior scientist at ICMR-NIV and director in charge of the National Institute of One Health. Their talk is titled Post-Pandemic Genomic Surveillance Advances, Helping the Pathogen Discovery and One Health Approaches in India. Dr. Varsha?
Thank you, Darcy, for introduction. And I would like to thank organizer for giving me chance to share my experience for pandemic preparedness and pandemic mitigation and post-pandemic surveillance system in India. So this is my disclaimer. For the COVID-19 pandemic response, how India has prepared themselves for catering such a huge population. So we, being a national influenza center and a reference laboratory for WHO, we quickly developed our own in-house real-time RT-PCR kit and got validated using WHO controls. This way, we have started our testing protocol in the country in as early as from 22nd of January, when entire world was just started thinking of like it is a pandemic now and we have to act on that. So what we did basically, we have standardized the protocol at NIC. We quickly made the CRA, the case referral forms, case definitions was identified, and this was disseminated to the national network of viral diagnostic laboratories, which are based in government medical colleges. So that's how we decentralize the testing protocol. We have shared the SOPs. We give the online training to all the laboratories to make sure that they are following an exact same protocol what NIC is following. We also shared the reagents what we made at in-house, the standardized kit, basically RT-PCR kits, to all these network partners. Until March, we have almost stayed than 125 laboratories across the country, and that's how we have enhanced our testing capacity in the country. After that, in 2021, there was a need for having the genomic surveillance network in place in country and to monitor the evolution of the SARS-CoV uh, virus. And we till that time we saw that second variant of SARS-CoV was already evolved, and we need to keep an eye on circulating variants in the country. So that's how this INSACOG, is Indian SARS-CoV Genome Consortium, was established, and this was jointly established by Ministry of Health and Family Welfare, Department of Biotechnology, CSI Scientific Research, and ICMR, Indian Council of Medical Research. And it has a hub and spoke model. Basically, there were 10 hub laboratories, and these hub laboratories are being attached with multiple laboratories as a spoke. So for ICMR, Indian Council of Medical Research, we are an apex laboratory, and with us there were 45 laboratories attached as a spoke laboratories, and they all were having exact protocol or SOP. We train them for SARS-CoV genome sequencing, how to proceed with different platforms, whether it is Illumina, ONT, or IM2Run platform. But mostly they have used Illumina platform because it was already established in the laboratory, and they were using the COVISEC protocol for SARS-CoV genome sequencing. We shared the SOPs, and also we did the centralized procurement of these reagents so that there will be a uninterrupted supply of these reagents to the laboratories. And that's how the data was collected at NIV Pune and that's how we build up the database for Indian SARS-CoV genome sequences in the country. And this was then submitted to GSAID platform from NIV Pune. So the data was shared to the policymakers, the laboratories, the governments, state governments, so that everybody are aware of what variants are in circulations and what is their impact on public health. So coming to the next, when there was a need of integrated disease surveillance, and WHO also emphasized that countries should have integrated disease surveillance for influenza, monitoring influenza and SARS-CoV because of their similar clinical manifestation. So we then quickly adopted this COVISEC protocol for basically to monitor influenza, RSV, and HMPV viruses, which basically influenza viruses mainly because of their seasonal and pandemic potential, and we have to keep an eye what kind of clade is in circulation for influenza viruses. For RSV, we have to have, like, it is basically a whole genome sequencing, which add in a knowledge that what kind of circulation pattern we are observing in the country, whether it is RSV-A is dominating or RSV-B is dominating, and which clade is in right now circulation. Similarly, HMPV and other important respiratory viruses such as adenovirus also we tried with repurposing of this COVISEC protocol. So it is basically the same protocol was followed, like it is a common workflow for NGS. It's RNA extraction followed by library preparation, and then on machine it will be get sequenced, the data will be generated, raw data is then mapped with the reference genome, and that's how we could collect the data. Now we could generate the whole genomes for major respiratory viruses, and this whole process was based on amplicon based sequencing. Basically we have identified the primers for amplifying entire genome for influenza, RSV, and HMPV. These are the kind of examples for how we have generated amplicon based PCR products, and these were then further using the COVISEC protocol we have then sequenced. So we have repurposed COVISEC protocol for getting the genomes for influenza, RSV, and HMPV. And this we found that it is basically a low cost, and we can scale up this sequencing for other respiratory viruses, which will be a very cost effective. We have used the CDC IRMA pipeline, bioinformatics pipeline for generation of and analyzing the consist sequences from raw data which is generated through sequencing. End-to-end workflow can efficiently sequence the full genomes of influenza A, B, and RSV and other respiratory viruses. So this is the example for how we have established entire surveillance as well as genome network in country. In a upper portion of the slide, you can see that we have this pan-India respiratory ILI-SARI surveillance network currently functional for almost 45 VRDL laboratories, and we are generating data, continuous monitoring of both these influenza and SARS-CoV viruses in the country, and this data is being fed to the WHO FluNet platform for global analysis. Similarly, in SACOG network is generating the genome sequences for SARS-CoV basically, and we are seeing all the variants in country, right from Delta to currently circulating KP1 and KP2. So you can see the different time point, different lineages or different variants of SARS-CoV in circulation. And till date also we are still monitoring SARS-CoV evolution in the country, and it is been also shared with global database like GSAID, and for country who are the data is been shared with the policy makers to understand the impact of these variants in public health. So with that, I am happy to take any questions. Thank you.
Thank you, Dr. Varsha, for providing such a beautiful narrative of the COVID work done by uh, ICMR-NIV team, but also with the VRDL team, which you just described, together how we could use the ISACOG in the country for enhancing our genomic surveillance. It was not only on the human specimen, but also on the environmental specimen through the waste um genomic sequencing, which actually paved the way to think. And the COVID crisis brought the concept globally that One Health approach should be used. And I'm going to narrate some of recent understanding how One Health approaches can be used in the country to enhance the surveillance, virus discovery, and improve the surveillance in India. In 2022, National One Health Mission for India was launched on July 2022, where 13 different departments are working wonderful in their own mandate, but ensuring synergetic across sectors is the key. The aim is to not duplicate any of the individual mandate and work of each ministry, but to bridge the gaps identified by providing focus components for intersectoral component. And this is just a beginning to understand how together we can achieve better, how we can be prepared better, and how we can improve the research and development in the country. This is a pictorial presentation which describes that how livestock health, human health, wildlife health can work together by bridging together with the different sectors to enhance the capacity building, making the networks, and moreover, how we can improve the surveillance to detect any unknown pathogens which can lead to the outbreak and pandemic. And as we all know that in any outbreak, identification of the pathogens through the next generation sequencing become very essential tool which everybody realized during the pandemic. And these lead not only the knowing about the pathogen, but help in making new vaccines and creating a lot of knowledge which can be translated further in the research. I'm going to share one of the our story, which is linked with the Dr. Varsha's narrative that how we could repurpose the COVID sick kit to use for the Casanur forest disease in India do the genomic surveillance. Casanur forest disease was discovered in 1956, 57, and it is a risk group category 3 pathogen and pose a significant public health concern with high mortality. And it is a zoonotic disease where non-human primates vector is ticks and humans are the end host engaged, and human get tick bite and get infected. And the outbreaks has been reported. National Institute of Virology made inactivated vaccine in past, which was implemented and used for years, but lately it was realized that it is not effective anymore. That bring a lot of questions to the enhancing genomic surveillance to understand if the disease has any change impact due to the virus mutation. And we started doing genomic surveillance across the Western Ghats. So these are the area which you can see. These are the 5 states which is affected by this disease. In 2024, there was a huge outbreak which we could record 231 KFD cases that included 12 deaths. Not only that, genomic surveillance and sequencing also help in understanding that existing molecular and serological test methods are still working with the mutation of the virus. But this also created the need of having more laboratory, and a VHF laboratory network are formed among these affected area, which they can immediately do the testing, provide a diagnosis. But these data, how we have used, I would come in that later. But before that, I would like to quickly describe how we use the Illumina Microbial Amplicon Prep for KFD whole genomic sequencing. as we know that this was used for COVID widely across the globe, but we by designing a very specific primers for the KFD virus, where we use the pool 1 and pool 2, and these primers by using, we did the PCR, and then we use the same method as we do with this kit. We have the step 3 where we put the indexes, and then we load in the machine, and we get the data and we analyze it. By this method, using this microbial amplicon prep, we could retrieve a beautiful sequences from the clinical sample, and which is immediately, very shortly, we can report, and we can assure that the mutations are not very high, and the sequences are matching with the earlier viruses which is circulating in Western Ghats. And if you know about Casanova forest disease, it's a flavivirus which has and we could see only less than 3% differences among these strains. And most of these clades are divided into clade 1 and 2. The recent strain come in the clade 2, and we could retrieve very quickly using this kit, and the recovery was very good. Even the CT value of 32, we could recover the sequences, and which really not only help in designing the new vaccine, but if we are going to make inactivated vaccine, or if we are going to do any further analysis using bioinformatics approaches, it can help to understand. It can also enhance and improve the KFD surveillance. Also, using this knowledge, we can develop new PCR, and we have already validated different PCR point of care. We could also validate our ELISA with the different strain, and we can assure that existing system would work. This also help us to understand the algorithm of testing method, and I'm sure that all these information will formulate to have a very good vaccine for KFD soon, which we will utilize in the future. With this, I thank all of you for giving this opportunity, and I will be happy to answer any question. Thank you very much.
Dr. Varsha, Dr. Pragya, thank you so much for sharing your experience. The National Institute of Virology in India is clearly a leader in addressing the challenges that are posed by emerging infectious diseases. We only have time for one quick question for both of you, but if you could offer one key piece of advice to other labs worldwide who are looking to enhance their preparedness and response capabilities for genomic surveillance, what advice would you offer? Dr. Varsha?
Well, with our experience, we can say that like decentralization of whether it is a testing or whether it is a genomic sequencing will give you a quick answer basically. To cater such a large number of population of India, that was actually key question how only NIV Pune can cater all this cater for the nation. So that was our good move that we have decentralized the testing as well as genomic surveillance. Secondly, in the era of integrated surveillance, basically to monitor multiple respiratory viruses which has a similar clinical features, repurposing of COVID platform protocol is helpful. That we have shown with our own data that we can generate the data full genomes for influenza, RSV, and HMPV, even adenoviruses. So these are the key viruses which needs to be again monitoring. So that will be my suggestion that we can repurpose very easily OBSIC plan. Thank you.
Dr. Pragya?
Yeah, thank you, Darcy. Dr. Varsha has given all very beautiful points. I would like to add that intersectorial coordinations and cooperation would help to understand the diseases in better cohesive manner. Rather, we are focusing one particular type of samples and one particular type of surveillance. So, one health approach should be key learning which we did from COVID. And to utilize that, we have to not only learn by each other in different sector how they do process the sample, how they do the sequencing, and chip in to make the course corrections, have the uniform SOPs, and I think capacity building would be the key for future pandemic and outbreaks for the novel discoveries of pathogens. So this will be my message to the rest of the people.
Great. Thank you both for that insight, and thank you again for your presentation today. Our next speaker is Dr. Felipe Naveca, head of the Arbovirus and Hemorrhagic Virus Laboratory at Fiocruz in Brazil. He also leads the National Regional Reference Center for Arboviruses and Monkeypox.
Hello, Darcy. Thanks so much for the invitation to be here with you. So first, I'd like to thank my colleagues from Illumina for this kind invitation to be here in this meeting. So I will talk about our experience using IMAP for emergent and emergent virus surveillance in Brazil. So first of all, what is genomic surveillance? Genomic surveillance goes beyond just sequencing pathogens we use, but is, of course, this is the main information that we have. Use this to monitor disease, to control pathogens, to tailor interventions, to develop countermeasures. And of course, these are very important information that we work together with other information that we have in surveillance. But one of the most important points related to genomic surveillance is that we have not only have very good information in refers to quality, but in a timely manner. If it's not timely, we cannot use this as countermeasure, for example. So we have been using Illumina Microbial Amplicon Prep in our laboratory for different pathogens. It's very straightforward protocol, so we can use this for different pathogens, for DNA or RNA pathogens in our Illumina benchtop sequencing systems. And in our experience, we have been using for runs with 384 samples in NextSeq 1000 or 240 samples in MySeq. For example, what we do to use this protocol, we do primary design based on the alignment of the pathogens that we are interested on. And then we use a tool called Primer Scheme to design those primers. But we also include primers designed using another tool like Primer3 to design primers for regions that we have many mutations. So we use a combination of primer and Primer3 to design those primers. And then I will show you some examples that we had successfully worked with this. The first one is the design primers that we use for this measles emergence in Brazil that occurred in 2028. In fact, the first genome that we did for measles was in a standard-based sequence, but we designed those protocols to be used on NGS, and we were able to recover the entire genome of different samples of measles genotype D8 using this protocol. At the time that we used this, we have just a few measles completed genomes to compare, and none from Latin America. And then I think that measles would be, we should use IMAP to sequence the measles to improve this virus surveillance for this very important virus. For example, this is what we did in this design. We had the measles virus D8 reference sequence, and then we aligned it with other full genomes that were available in GenBank, and then we designed amplicon-based strategy for that. And I'm showing you here on the bottom genome that was recovered from from this strategy covering the entire almost the entire genome of this virus. And then I will move to another example that is dengue 2, genotype 2, the cosmopolitan genotype that emerged in Brazil in 2022. In this paper, we published a study where we sequenced 237 genomes from those dengue 2 from different places in Brazil, and most of them were sequencing using also an IMAP-based strategy. So with these results, with completed dengue 2 genomes, we were able to reconstruct the introduction of this genotype in Brazil, and we could see that at least 4 times this virus was introduced in Brazil. And then this was very fast using this strategy, and we could have very good results. Here is an example of this genome assembled with this strategy. We could recover almost the complete sequencing of this virus using this approach. Another example is this what we did when we detected the first dengue serotype 3 cases in Brazil in 2023. In fact, our colleagues in Roraima state were the first one to detect the presence of this virus, and also we have colleagues from LACEN, Paraná, that also detected one imported case of this virus. And it called our attention because dengue virus serotype 3 has not been circulated in Brazil for over 15 years. So it was necessary to characterize this lineage, and we did it very fast, also using protocol based for that. The first sequence that we generated were using Illumina VSP, another strategy, but after that we designed primers and probes to reconstruct to sequence the dengue-free genome using amplicon-based approach. So I think that the combination of both is important when we have a new virus circulating. So with this result, we could see that we found a new clade among the genotype 3 of the dengue serotype 3, and then we could, using phylogeographic approach, we could show that it is a new agent delivered from Asia, but as a new lineage in Brazil that was detected, but not only in Brazil, also in the Americas, and we called it dengue 3 genotype 3 American 2 clade. And it's interesting, this virus is now circulating in different places, as this paper is showing the detection in the United States, in Florida, and they identified the same genotype that we have already described. And the same was in this paper from São Tomé and Príncipe, where our colleagues could also identify the same genotype that we previously described. So, as I told you, the first genomes were sequencing using VSP, but after that, after we have those genomes as reference, we designed an amplicon-based approach using IMAP, and then we have now some the other genomes that were recovered using this strategy. As you may see here, we can also sequence almost the entire dengue-3 genome using this strategy. We did the same for dengue 1. Dengue 1 is still a very important arbovirus in Brazil. This serotype is still causing a lot of cases in Brazil. And then for several years, we have genotype 5 as the main one. And then we designed a new approach, an updated approach using IMAP for genotype 5, dengue 1 genotype 5. And then we also recovered the entire genome for this virus. I'd like to show you another example with an emerging arbovirus that circulated in Brazil. First of all, we will introduce this virus. It's called the species is Orthobuniovirus oropochense. The prototype of this species, the Oropushi is a virus linked to acute febrile outbreaks and also encephalitis. But recently, the first deaths and the first case of vertical transmission with the fetus death was confirmed in Brazil. So this virus is emerged since the last year in Brazil, and we had also an outbreak caused by this virus in Brazil, and this virus is now being detected in different countries, for example, in Bolivia, Peru, Colombia, and the same lineage that we described is also being detected in other countries like those, as said, including some imported cases were reported in Europe, like Italy, Germany, Spain, and in Central America we have now an outbreak in Cuba. So this virus is kept in nature in two cycles. One sylvatic cycle that we have humans, non-human primates, rodents, and sloths as the main reservoirs. But we also have what we call the urban cycle, where we have human-to-human transmission, but not directly. The transmission is the vector for this transmission urban cycle is Culicoides paraensis. So it's another vector, it's not Aedes aegypti. So we have some different aspects to consider in these arboviruses. But we also have information that Culex kinkefaciatus may also act as a secondary vector. And then we did quite the same that we had for dengue 3. The first one we sequencing using shotgun metatranscriptomics using Illumina Ribble 0, a sample that we had isolated in our laboratory. And then based on this unbiased genome, we designed an amplicon-based approach to use it for other samples. And then, as you may see here, forgot to highlight that this virus is a segmented virus. Then we designed primers for the 3 segments. Here I can show you the L segment covering 98.3% of this segment. For the medium segment, we have almost 99% covered by this strategy. And for the small segment, we have 87% sequencing using this approach. And for this segment, it's also important that we have the diagnostic target that we are using in Brazil and different other countries in Latin America. It was a protocol that we designed a few years ago. And then we have the information of those cases in Brazil in this preprint that is now under review. So if you want to see more details, please go to this preprint. And then I will show you, just to highlight what we did using IMAP. We were able to sequence genomes from this region in Brazil. In this map, I will show you the municipalities where the samples were collected and sequenced. There on the right, we will see the phylogenetic context, and on the bottom, we may see the epidemiological weeks and the case that we have. So as you can see here, we sequenced from different municipalities in Brazil for since 2022 to 2024, and then we could see that we had an outbreak in 2023, but into the end of 2023 in the early 2024, we had much more cases in Brazil. So you can see here that we were able to, using this strategy, to sequence almost 400 genomes from 36 municipalities from 4 Brazilian states over 77 weeks. So this was a very important information that helped us to better understand how this virus is spread over Brazil. And what other important point that we could see is that the current outbreak is caused by a new virus that we call a reassorted virus. that emerged from different reassortment events from lineage that circulated in the past. In fact, we had at least 3 events of reassortment. It's a phenomenon where the virus, 2 different viruses may infect the same cell, and we have a new virus that has part of the genomes of virus A and virus B, and then this is a new virus. And then we could, using the sequencing protocol, we were able to show you that we have a new emergent virus that emerged from different reemergent steps that occurred in the past. In this emergent virus is now spread not only in Brazil, it's the same lineage that is circulating in different countries in Latin America. And also that the cases that were identified in US and Europe. So I would like to show that what is going on with this outbreak in Brazil. As you may see here, we had most of the cases occurred in the rainy season. So of course, this is a virus transmitted by a vector, and then it has a very important relationship with the weather. So this vector grows when we have a rainy season in organic matter for its reproduction. And then here you may see that we had in the very end of 2023 and the beginning of 2024, almost all cases that were suspected for arbovirus infection in the Amazonas state were caused by Europush. In this picture, I may show you that we have different clades emerging from this new lineage. We call the main ones a macro, is this region here that is in the border with different states in the Amazon region in Brazil, and another lineage that is circulating only in the Amazonas state. And also using this approach, the sequence approach, we are able to identify mutations that are occurring on the surface of this virus on the proteins that attached to the viral host, to the cell host, and then the virus is to enter these cells. So we can also use this information to see how the virus is evolving in those important antigenic sites of this virus. Other result that we could see using this sequencing approach, and of course epidemiological information, is that we see that the virus population increased a lot during the rainy season, then it decreased, and then rise again, increases again in the rainy season in the end of 2023. The same for the different clades, as you may see here in red and in green. The different clades, all of them increased during the rainy season. And we are also able to reconstruct the spatial velocity of this virus using this genomics-based approach. So we could see that the virus was spreading following the spread that we expected for a virus transmitted by vectors, and most of those migrations were that we consider small range migrations that is in agreement with a virus that is spread by vectors. So since we have different areas in Brazil that are not endemic and are reporting this virus, this was also an important result that allowed us to better understand how this virus was spreading. And then also based on the genomics results that we got, we were able to reconstruct the pathways of this virus spreading in different places in Brazil using a continuous phylogeographic approach, as the virus was spread in the very beginning, and from 2022 and 2023 to 2024, and the other is clade in the Amazonas state circulated in 2023 and the beginning of 2024 when it spread for different municipalities in the Amazonas state. So we have a lot of things that we can recover using genomics and also epidemiological information. And we are glad that this virus is not so neglected anymore. We have some reports calling attention for this virus, like this one that was published in Science in June. And there is another one that was also published in Science in July, when the first infections in fetus were suspected, and then now we have those confirmed, and we have another virus to be studying in the next years. So with the genomic surveillance, we were able to recover much more information than we had using other tools for classical epidemiology. So our group is working in this virus discovery and emerging virus information for years, as in this study that was published, that in this news that was published in Science. So I'd like to end this to say thank you for all the partners and grants that we received to conduct our studies. And I'll be glad to take any questions if you had. Thank you again for the opportunity to be here.
So Dr. Naveca, we have time for just one question for you. Given Fiocruz's success with amplicon sequencing with IMAP for rapid outbreak response and surveillance, it's really remarkable. What are some key practices that other labs could adopt to achieve similar results?
The first point is IMAP is really easy to use. So we have been using this since it was formerly known as COVID-seq, and now we started using in the very beginning of the pandemic. In fact, our lab was the first one in Brazil to use that. So we were so successful using this approach that we started testing for other pathogens. And then what I could say that was like a tip for other researchers is just to use fresh material. I mean, those that you conducted the virus extraction, RNA extraction, especially if you are working with RNA, and then following the primary design using those protocols that I showed you, like primer scheme and primer tree. It will be easy to use this protocol for other different pathogens. So we are very glad to use IMAP in our routine.
Great. Thank you so much for your insight and support with this. I'd like to thank our speakers again for such an informative webinar. I'm sorry we weren't able to address all of the questions that we've received, but we have recorded those and we will be publishing answers soon. This webinar really highlighted the need for flexible pathogen genomic surveillance to support public health efforts, and highlighted how the IMAP kit can really help support those efforts for any virus of interest. I'm going to bring our host back now to close things out.
Thank you, Darcy. And thank you to Dr. Varsha, Dr. Pragya, and Dr. Naveca for this very informative webinar and for sharing their valuable insights on genomic surveillance. Now, before we go, I do want to thank the audience for joining us today and for their interesting questions. Due to time constraints, we apologize that we were unable to address all of your questions during today's webinar. Additionally, you may continue to submit questions during the on-demand period, and we will review those questions and do our best to provide responses. Today's webcast can be viewed on demand, and Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. If you haven't already done so, we invite you to partake in our survey. And for your convenience, the survey will pop up for you when we close. Your feedback is vital in helping us provide you with the most relevant technical resources following the webinar. Thank you again for joining us. Until next time, friends, take care. Bye-bye.